In the before class, we have seen the oven drying method in the moisture analysis, that is water analysis. Now, we will see the distillation method. The distillation method is a classical technique used in the food analysis to determine the moisture content of the samples, particularly those containing the volatile substance. Volatile substance means that can be easily vaporized or when water is present in the small amounts. This method involves separating water from the samples through co-distillation. What is co-distillation? Co-distillation is the distillation process that is performed on mixtures in which the two compounds are not miscible with an immiscible solvent such as toluene or the xylene. These are the organic solvents which does not mix with the sample. Coming to the principle is based on the co-distillation of water with an organic solvent that has the higher boiling point than the water, but they do not mix with each other. As the mixture is heated, water vapor and the solvent vapors are formed. These vapors are then condensed and the water is separated and measured. Here they form the azeotropic mixture, means it is a combination of two or more liquids with similar boiling point and the same composition in the vapor and the liquid phases. Even though they have the same boiling point, they do not mix with each other because they are immiscible with each other. So, in the trap we can see that two layers are formed. Now, coming to the procedure. First, we will prepare the sample. Place the food sample in a distillation flask along with an appropriate amount of an immiscible organic solvent, that is toluene or the xylene. Next is distillation. Heat the flask. The water and the solvent will form an azeotropic mixture and vaporizes together. Condensation and collection. The vapors are passed through the condenser where they cool and condense into a liquid. The condensate is collected in a graduated receiver where water and solvent separated due to the immiscibility. Measurement. The volume of water is measured directly from the graduated receiver. This is the Dean-Stark method. It is a technique that is used in the chemistry to remove water from the reaction mixtures, and it is employed in the reactions where water is the byproduct, such as esterification, dehydration or condensation reactions. The Dean-Stark apparatus consists of a round bottom flask where the reaction occurs, a reflex condenser and the trap, are also known as the receiver. This is the receiving flask, and the trap is calibrated to measure the volume of the water collected. Here we can see the condenser, it is connected to the pipe and another end it is connected to the sink, so that the cool water enters into the condenser and it carries the heat from the condenser and it allows to the sink to flow out. This is the outlet of the pipe and this is the inlet of the cool water into the condenser. This condenser removes the heat from the gas or the vapor to condense into the liquid. After condensation, we get the liquid drop by drop into the receiving flask. This reaction mixture is heated to its boiling point. Usually, this mixture consists of an organic solvent that forms an azeotrope with the water. The solvent and water in the form of azeotropic vapors rises up and pass into the condenser, where they cool and condense back into the liquid. Next is the trap collection. The condensed liquid, water and the organic solvent flows into the Dean-Stark trap. Since the water and the solvent are immiscible, they do not mix. They form two separate layers here. The denser water settles at the bottom of the trap while the organic solvent, which is less dense, remains on the top. Here we can see the water. Here upper layer we can see the organic solvent. The organic solvent is often recycled back into this reaction flask for continuous use, while the water accumulated in the trap and it is removed from the system. The water is effectively removed from the reaction, driving the equilibrium of the reaction forward. This method is valuable in organic chemistry and the industrial processes where precise water removal is essential. Applications in food analysis. The distillation method is particularly useful for analyzing samples with volatile components or oils such as spices, essential oils, flavorings where water content needs to be accurately measured without interference for the other volatile substances. Advantages and limitations. It can measure water content in the presence of volatile substances, suitable for samples where other moisture determination methods are not applicable. Limitations is it's time consuming and requires careful handling of the organic solvent, less precise than some other methods like Carl-Fisher titrations for samples with very low moisture content. This distillation method is useful technique in food analysis for specific types of samples, offering a reliable way to measure water content when other methods may not be suitable. Brown dual distillation method. It is a well-known chemical method for the moisture determination, particularly in the grains and other agricultural products. It involves the use of the solvent distillation method to separate moisture from a sample. This method involves heating the sample in the presence of a solvent and then collecting the distilled water to measure its amount. It was commonly used before more modern techniques like oven drying or Carl-Fisher titration. Now coming to the procedure, sample preparation. The sample, that is grain or any food product, it is placed in a flask and a suitable organic solvent, often xylene or toluene, is added. A solvent, typically xylene or toluene, is added to the flask. These solvents are chosen because they form azeotropes with water, allowing water to distill at lower temperatures than its normal boiling point. Next is the heating. The mixture is heated, causing the water in the sample to evaporate. The solvent helps to co-distill the water. Condensation. The vapors of the solvent and water rise through a condenser where they cool and condense. The condensed mixture then flows into a graduated receiving tube. Next is the separation. Since the solvent and water are immiscible, they separate into two distinct layers in the receiving tube. The water, being denser, settles at the bottom and its volume can be measured directly. Measurement. The volume of water collected is noted and from this the moisture content of the sample can be calculated. Here we can see the heating lamp, mineral oil, distillation flask, thermometer, hot water bath and the graduated cylinder. Here this is the sample where a known weight of a seed or grain or mineral oil is placed in this distillation flask. This is the solvent. This mineral oil is mixed in this solvent where it is a xylene or toluene. This forms the azeotropes with water, allowing water to distill at lower temperature than its normal boiling point. And now the mixture is heated and the water in the seeds or grains evaporates along with the solvent, forming a azeotropic mixture. The vapor is then passed to the condenser where it cools and turns back into the liquid. The condensed liquid consists of both water and solvent. It is collected in the graduated cylinder. This is also called as trap. Since water and the solvent are immiscible, they form two distinct layers. The water, being denser, settles at the bottom of the receiver. The volume of water collected in the trap is measured, which gives an indication of the moisture content in the original sample. This method is now less commonly used in favor of the faster and the safer techniques, but it still has the historical significance in the moisture determination in the older days. Coming to the advantages and application. Precision. It is precise and can accurately determine low levels of moisture content. And it is particularly useful for materials where moisture is tightly bound or where other methods may not effectively release or measure the water content. Chemical nature. Being a chemical method, it is especially suited for the substances that may decompose at the temperature used in these methods like oven drying. This method is a robust or reliable way to determine the moist content, particularly in samples that are challenging to dry completely or where bound water needs to be accurately measured. And the disadvantages are that it is time consuming and use of organic solvents which are hazardous and pose health and environmental risks, and we should have a specified distillation equipment such as condenser, receiver, etc. Now coming to the chemical methods. It is used for determining the moisture content in food analysis, often involves reactions that quantify the water content in a sample. Here are some common chemical methods. Carl-Fisher titration. It is more specific method for determining the moisture content, particularly used for the samples with low moist content or those containing volatile substances. This titration is precise analytical technique used to determine the water content in various substances including the food products. It is especially valuable for measuring the low levels of moisture, and it is effective for samples where water content is tightly bound or present in the small amounts. Coming to the principle, the Carl-Fisher method uses a chemical reaction that specifically measures water content. It involves titrating the sample with the reagent until all the water has been reacted. It is based on the quantitative reaction of water with the iodine in the presence of the sulfur dioxide and a base which is called a pyridine or the imidazole in a suitable solvent, often methanol. The chemical reaction involves the water, iodine, sulfur dioxide and the base to form this complex. And Rn is the organic base, that is pyridine. This iodine is added from a burette and the amount of iodine required to react with water in the sample is measured. The volume of iodine titrate used is proportional to the amount of water in the sample. Water reacts with iodine to form the hydriodic acid, and once all the water in the sample has reacted, the titration ends, allowing for the precise measurement of the water content. It is best for the low water content samples, typically less than 1% water content. Carl-Fisher titration remains one of the most trusted and precise methods for moisture analysis across a variety of fields. I have already explained this equation and everything in the objective methods of food analysis. If you have any doubt, you just see in the second video. Applications in food analysis. Accurately measures moist content in foods with low water content such as oils, fats, powdered foods and dehydrated products. It is also useful for the samples containing the volatile substance that might interfere with the other moisture determining methods, and it specifically measures the water content. It is highly accurate, sensitive and can be automated, making it its preferred choice in quality control and the food safety assessments. Next is the chemical desiccation. Principle is the desiccants are the chemicals that absorb moisture. In this method, a desiccant is used to absorb water from the sample. The chemical desiccation in moisture analysis refers to the process of using chemical substances. They are desiccants to remove moisture from a sample. This technique is often used in the laboratory settings to ensure accurate moisture content determination. It is crucial in industries like food, pharmaceuticals. Water desiccants. These are the chemicals that absorb or adsorb moisture from the sample. Some common desiccants are calcium chloride, CaCl2 and silica gel, and phosphorus pentoxide P2O5. These are the common desiccants that are used in the food industry. Procedure. Sample exposure. The sample is exposed to a desiccant like anhydrous calcium sulfate or the phosphorus pentoxide. Weigh measurement. The desiccant absorbs moisture from the sample. The moisture content is determined by the increase in the weight of the desiccant or the decrease in the weight of the sample. Chemical desiccation helps in determining the total moisture content of a sample by drying it completely. The sample is placed in a container along with the desiccant that will absorb all the water, ensuring that the sample becomes completely dry for an accurate measurement. The sample is first weighed and then placed in a closed environment with a desiccant. After several hours, the sample is reweighed. The difference in the weight before and after drying gives the moisture content of the sample. The desiccant needs to be replaced or regenerated periodically because it becomes saturated with water over time. What are the advantages? It is useful for the sensitive materials that cannot be heated, which is common in thermal drying methods. Low temperature operations ensures that volatile components in the samples are preserved. Applications. It is suitable for the foods that might lose volatile substances at the higher temperatures. This chemical desiccation is ideal when heat based drying methods like oven drying which alter the sample's properties, or when the sample contains compounds that decomposes upon heating. In those cases, we use the chemical desiccation method. Next is the sorption isotherm method. Coming to the principle, this method involves measuring the water activity of the food sample and using it to determine the moisture content based on the sorption isotherm curve. This sorption isotherm method in food analysis, it is a technique. It is used to study the relationship between the moisture content of the food product and the water activity at the constant temperature. It provides critical information about how a food product interacts with the moisture, which is important for understanding its stability, shelf life, texture, and overall quality of the food product. The sorption isotherms are particularly used to assess the drying process and the packing requirements and the storage conditions. Here, the water activity is the measure of the free water available in the food for microbial growth, enzymatic reactions, and the chemical reactions. It is measured on a scale of 0 to 1. 1 represents the pure water and 0 represents the complete dryness. The lower the water activity, the longer the shelf life, as the microorganisms cannot grow in the low water activity environment. Water activity represented as AW. Next, coming to the moisture content. It refers to the amount of water present in the food product. Usually it is expressed as a percentage. Next, coming to the procedure, sample equilibration. The sample is equilibrated at a specific related humidity. The specific related humidity is the amount of water vapor in a unit of moist air. This relative humidity is the percentage of the moisture in the air compared to the how much moisture the air can hold at a given temperature. Next is a measurement. The water activity is measured using a water activity meter. Moisture content calculation. It is determined using the sorption isotherm curve, which relates the water activity to the moisture content. A sorption isotherm curve, it gives the relationship between the moisture content of the food product and its water activity at a constant temperature. This curve helps in understanding how food absorbs or desorbs water under varying humidity conditions. This curve is essential in food science because it provides a moisture interaction which influences microbial growth, chemical stability, texture, and the shelf life. Now coming to the components of the sorption isotherm curve. Water activity AW, which is represented on the x-axis. Water activity represents the availability of the free water in the food for the microbial and the chemical activity. It ranges from the 0 to 1. 0 means completely dry and 1 means it's of pure water. As water activity increases, the food becomes more prone to spoilage and quality degradation due to the microbial growth or the chemical reactions. And the water content or the moisture content percentage is represented on the y-axis, which indicates that the total amount of water, that is free or bound water, that is present in the food product, often expressed as a percentage of the total food mass. Different foods have different moisture content levels, and the amount of moisture they can hold varies depending on the humidity of the surrounding environment. The sorption isotherm is temperature dependent, meaning the relationship between the moisture content and the water activity changes with the temperature. The curve is generated for the specific constant temperature. Adsorption isotherm shows how much moisture a food product absorbs when exposed to increasing humidity. This is relevant for foods during storage or in humid environment. This is desorption isotherm. It shows how much moisture is lost by the food as the surrounding humidity decreases. This curve is essential in processing like drying or when foods are stored in low humidity conditions. This sorption isotherm curve typically has 3 distinct layers. Region 1, that is bound water, strongly bound monolayer. At low water activity, AW is less than 0.2. The moisture content is very low and most of the water is tightly bound to the food's molecular structure, example proteins, carbohydrates. This water is not available for the microbial growth or the chemical reactions. Food in this range are stable with low spoilage potential. Now coming to the second region, that is the intermediate moisture. This is a transition zone where moisture content starts to increase with water activity, but water molecules are still interacting with the food matrix. Water is more available for the enzymatic and the chemical reactions, but microbial growth is still limited. Food in this region are less stable and may require protective packaging or controlled storage environment. Now coming to the third region, that is free water. At higher water activities, AW is greater than 0.6. Free water becomes available in the food. This region represents a critical point where the moist content rises rapidly with a small increase in the water activity. Foods are highly susceptible to microbial growth, enzymatic degradation and spoilage. This region is often avoided for long-term storage of most foods due to the high risk of spoilage. This monolayer moisture content is the key point on the isotherm where the food reaches optimal stability. At this point, water is bound in a monolayer around the food molecules, minimizing chemical reactions and the microbial activity. The monolayer is where the food has just enough moisture to prevent textural degradation without promoting spoilage or the unwanted reactions. Above a certain water activity threshold, that is around AW 0.6, most bacteria, yeast and molds can thrive well, leading to the food spoilage. This is the vital point for the food preservation strategies. This is called the sigmoidal curve. Most food products have a sigmoidal, that is S-shaped sorption isotherm. In this curve, there is a slow initial increase in the moisture content at low water activities, followed by a more rapid increase at the intermediate and the high water activities. These curves helps to determine how much moisture needs to be removed to ensure a food product is stable or it is safer for the longer term storage. The sorption isotherm curve provides guiding decisions relating to drying, storage, packing and the shelf life management. Coming to the applications used for the foods where precise moisture content and the water activity relationships are known. These chemical methods are essential for the accurately determining the moisture content in the various food products, ensuring quality control and the compliance with the standards. If you like the video, please do like, share and subscribe. Next is the gas production methods. The gas production methods in food analysis are the techniques used to evaluate the fermentation activities of the microorganisms. Microorganisms means bacteria, yeast, etc. are the chemical reactions that produces gases. These methods are essential for understanding the quality, safety and the functionality of the food products, particularly in determining the microbial activity, fermentation process, spoilage and leavening. Common gases measured here are the carbon dioxide, commonly which is produced by the yeast during fermentation, in bread making, or the bacteria during the carbohydrate metabolism. And another gas is the hydrogen and the methane. They are typically associated with the activity of certain gut or the spoilage bacteria. Next is the sulfur compounds, H2S, can be produced during the protein degradation or the spoilage. And another gas that is measured is ammonia, may be released during the protein breakdown. Commercial instruments are also available that utilize specific reactions between the chemical reagents and the water that leads to the production of a gas. Example, when a food sample is mixed with the powdered calcium carbide, the amount of the acetylene gas produced is related to the moisture content. The amount of gas produced can be measured by the volume of the gas produced, the decrease in the mass of the sample after gas is released, and the increase in the pressure of the closed vessel. Durant tube method. A small inverted glass tube, that is Durant tube, is placed inside the larger test tube containing a liquid growth medium inoculated with the microorganisms. If the microorganisms ferment carbohydrates, they produce a gas, that is usually carbon dioxide, which gets trapped in the Durant tube, forming a visible bubble. This is a technique used to detect the gas production by microorganisms, particularly during fermentation. It involves placing an inverted small test tube called as Durant test tube, which is placed inside the larger test tube containing the liquid culture medium. This method is commonly used in microbiology to assess the gas producing ability of microbes especially in tests for carbohydrate fermentation or in the study of bacteria like Escherichia coli for detecting coliforms. Here we can see the capillary tube, large test tube, small durum tube and the gas bubble formed here. A durum tube is inserted upside down in a test tube containing a growth medium, that might be sugar broth or the lactose broth, before sterilization. The medium is then inoculated with the microorganisms being tested. The tube is incubated under suitable conditions for the microorganisms to grow to metabolize the nutrients. If the microorganisms ferment the carbohydrates such as glucose or lactose, which produces the carbon dioxide, the gas will accumulate in the durum tube, forming a visible air bubble at the top of the inverted tube. If no gas is produced, the durum tube remains filled with the liquid medium. We can see here this is uninoculated, where gas is not produced, the durum tube remains filled with the liquid medium. Here lactose broth is present, the sample is incubated for 24 hours. We can see the air bubble formed in the durum tube. This durum tube method used to detect the gas production by the microorganisms during fermentation. It is often used in the microbiological testing for food and water safety and in the microbial identification. Coming to the applications, it is used to detect gas producing bacteria in the food products such as coliforms, which indicates the contamination. Example is detection of Escherichia coli, E. coli in water or the food samples. Next is the manometric method. It is a technique used to measure the gas exchange such as oxygen consumption or carbon dioxide production by monitoring pressure changes in a sealed system. This method is commonly applied in the biochemical and the microbiological studies to assess respiration rates and the metabolic activities in organisms or cells. This technique measures the pressure of the gases produced in a sealed container. A gas is produced by the microbial or the chemical reactions. The pressure inside the container increases, which can be measured using a manometer. Manometer means which measures pressure changes. This is the sealed chamber, which consists of the sample. Sample means it may be food, microorganisms or the enzyme solutions, which is placed in this sealed chamber connected to the manometer. Manometer means which measures the pressure. The chamber may contain a liquid medium and the system is designed to detect the changes in the gas volume or pressure as the sample respires. As respiration occurs, oxygen is consumed or carbon dioxide is produced. The manometer detects small changes in the gas pressure within the sealed system. These changes are proportional to the amount of oxygen consumption or the carbon dioxide produced, allowing for the calculation of the respiration rates. The manometric method is a powerful technique for measuring gas exchange in the biological and the chemical systems, commonly used in respiration studies and fermentation monitoring and enzyme activity analysis. Applications: often used in fermentation studies to measure carbon dioxide production, such as in bread dough fermentation or the production of the alcoholic beverages. It can also be applied to study spoilage organisms or the metabolic activities in the probiotics. Example is measuring the fermentation rate of the yeast in dough during bread making to optimize leavening. Leavening means a substance is used in dough to make it rise, such as yeast or the baking powder. Next, coming to the respirometric method. It is a technique used to measure the respiration rate of microorganisms or the biochemical process in the food by monitoring gas exchange, particularly oxygen consumption and the carbon dioxide production. This method is commonly used to study microbial activity, fermentation and the spoilage process. And this method measures the respiration of the microorganisms by tracking oxygen consumption and the carbon dioxide production. It is monitored in a closed system, and the rate of the gas production is used to measure the microbial activity. A sealed system is set up where the food sample or the microbial culture is placed. As microorganisms respire, they consume oxygen and produce carbon dioxide. The respirometry measures the changes in the oxygen level or the amount of carbon dioxide released over time. The rate of respiration is then calculated based on these changes. This shows the microbial growth or the metabolic activities in the food. This NaOH pellets, that means sodium hydroxide, that is used in the respirometric method to absorb carbon dioxide produced during respiration. The reason for this is to allow accurate measurement of the oxygen consumption without interference from the carbon dioxide produced by the sample. And the magnetic stir bar is used to mix the sample continuously, and the sample is present with the bacteria, and the pressure sensor is present. And this pressure sensor is used in the respirometric method to monitor changes in the gas pressure within a sealed system. These changes reflect the consumption of oxygen and the production or absorption of the carbon dioxide during the microbial respiration or the biochemical process in the food sample. The respirometry method is a valuable tool in the food analysis for monitoring microbial activity, fermentation and spoilage by measuring the gas exchange, offering the food quality and the safety. Coming to the applications used to assess the metabolic activity of the microorganisms in the fermented foods such as dairy products, fermenting vegetables or the probiotics. And example is monitoring the metabolic activity of the lactobacillus in yogurt production by measuring the carbon dioxide. Infrared gas analyzers IRGA. This is used to measure the concentration of the gases like carbon dioxide and methane produced during the biological or the chemical processes by detecting how these gases absorb infrared radiations. This method is commonly applied in the study of gas production in food fermentation, respiration and the spoilage processes. These devices detect specific gases, typically carbon dioxide, by measuring how much infrared light the gas absorbs. The gas sample is passed through the infrared beam and the amount of light absorbed corresponds to the concentration of the gas. This infrared gas analyzer is commonly used to study the gas exchange in live plants, particularly for measuring the photosynthesis and the respiration rates. It works by detecting changes in the concentration of gases like carbon dioxide and water vapor in the air surrounding the plant leaves. Here, the plant takes in the carbon dioxide during photosynthesis and release oxygen. They also release carbon dioxide during respiration. The IRGA measures the concentration of CO2 in the air before and after it passes through a chamber containing the plant or the plant leaves. The reduction in carbon dioxide level due to photosynthesis or increase in the carbon dioxide level from the respiration process is quantified by the IRGA based on how much infrared light is absorbed by the carbon dioxide molecules in the chamber. During photosynthesis, plants absorb carbon dioxide from the atmosphere and converts it into sugars, releasing oxygen as a byproduct. An IRGA can monitor the reduction in the carbon dioxide concentration in real time, giving a direct indication of the photosynthetic rate. This is typically done by enclosing a leaf in a leaf chamber that is connected to the IRGA, where the gas exchange can be measured continuously. During periods when photosynthesis is not active, at the night time, plants respire releasing carbon dioxide. The IRGA can measure the amount of carbon dioxide produced, allowing for the study of the plant's respiration. Some IRGA systems can also measure water vapor levels, allowing for the estimation of the transpiration rates. Means the process by which the plants lose water vapor through their leaves. By measuring both carbon dioxide uptake and water vapor release, researchers can assess a plant's water use efficiency. Applications. Frequently used in the fermentation industry to monitor carbon dioxide production in the real time, helping to optimize processes like beer and wine fermentation. Example is real-time monitoring of the carbon dioxide level during the fermentation of beer to assess yeast activity. Hydrogen sulfide production test. This is a microbiological test used to detect the ability of the microorganisms, especially certain bacteria, to produce hydrogen sulfide gas as a result of breaking down sulfur containing compounds. It is commonly used in identification of bacteria, particularly those in the Enterobacteriaceae family such as Salmonella or Proteus species. This test is used to detect the presence of the sulfur reducing bacteria, which produces hydrogen sulfide gas during the breakdown of the sulfur containing amino acids in proteins. It often uses media that reacts with the hydrogen sulfide to form the black precipitates indicating the gas production. A bacterial sample is inoculated into the media using a sterile loop or the needle. The inoculated medium is incubated at a suitable temperature, which is normally 35 to 37 degrees centigrade, for 18 to 24 hours or longer time, depending on the growth rate of the bacteria. After incubation, means that is 18 to 24 hours later, the medium is examined for the presence of the black precipitate. If blackening is observed, it confirms the production of H2S gas by the bacteria. H2S positive gives the black precipitate. If the H2S gas is not produced, then the black precipitate will not form, a yellow liquid will be seen. This H2S production gas test is the method to detect the ability of bacteria to produce H2S gas from the sulfur containing compounds. The production of this black precipitate in the test media indicates the presence of H2S gas, which is valuable in the identification of the sulfur metabolizing bacteria, particularly in the food safety and the microbiological studies. Next, coming to the application, detection of the spoilage organisms or pathogens that produce H2S during food degradation. Example, testing for spoilage in fish or the meat, where H2S production indicates bacterial contamination or degradation. Pressure transducer method. This technique uses pressure transducers to measure the pressure change in a closed system as a result of gas production. The pressure changes are directly proportional to the volume of the gas produced by the microbial or the chemical activity. This is used to measure the change in the gas pressure in a closed system. A pressure transducer converts pressure changes into an electrical signals, which can be monitored and analyzed to determine gas production or consumption. It is often used in the microbial respiration or the chemical reactions. The sample, that is food or the biological material, which is placed in a sealed chamber where gas exchange is expected to occur. A pressure transducer is attached to the closed chamber to monitor any changes in the internal gas pressure. As gases are consumed, that means oxygen, or produced, that is carbon dioxide, the total pressure inside the chamber changes. The pressure transducer senses these changes in pressure and converts them into an electrical signal that is proportional to the gas exchange. The pressure data can be continuously recorded and analyzed to determine the rate of gas production or consumption. For example, a drop in pressure might indicate oxygen uptake, while an increase in pressure could suggest carbon dioxide production. The pressure transducer is calibrated based on known gas volumes and pressures, allowing accurate interpretation of the pressure change in relation to the amount of gas exchanged. It is a sensitive technique for measuring the gas exchange in the biological and the chemical systems. By tracking pressure changes in a sealed environment, it provides real-time processes like microbial respiration, fermentation, and enzymatic activity. This method is widely used in the food science biotechnology to monitor gas production or the consumption. Coming to the applications, widely used in food microbiology to assess gas production in anaerobic fermentation such as methane production in food waste or the biogas production. Examples are monitoring methane production from anaerobic digestion of food waste or slurry during the biogas production. The gas production methods are crucial in the food analysis for understanding microbial activity, fermentation efficiency, spoilage detection, and overall food quality. They help food scientists optimize production processes, ensures food safety, and enhance the product quality. Next, coming to the physical methods. In this, we will see the electrical methods. Dielectrical method. The dielectrical method in the food analysis is used to measure the dielectrical properties of the food materials, which are related to how food interacts with the electrical fields. These properties are influenced by the moisture content, temperature, and the composition like fat, salt, and the protein levels of the food. And the moisture is determined by the measuring the change in the capacitance or the resistance to an electrical current passed through the sample. Limited to the food contains not more than 30 to 35% moisture. The dielectrical method provides a non-destructive rapid way to assess the moisture and other properties in the food, making it valuable for the quality control and the processing. Next, coming to the conductivity method. The conductivity method in the food analysis measures the electrical conductivity of the food sample, which indicates its ability to conduct an electrical current through it. This property is influenced by the concentration of ions, that means salts, acids, bases which are present in the foods, as ions are the primary carriers of the electrical charge in liquids. The salts and mineral content helps in conductivity. Conductivity is commonly used to measure the salt levels in the food like dairy, meats and the processed products. Salts increases the electrical conductivity. During fermentation, changes in the ionic concentrations can be tracked using the conductivity to monitor the progress of the fermentation in products like cheese, yogurt and pickles. And it also used to assess the purity of the water used in the food production, as dissolved ions affects the conductivity. The conductivity method helps to measure the ion concentrations in the food, making it useful for the quality control and monitoring the fermentation and the salting. Next is the hydrometry. Hydrometry in the food analysis involves using a hydrometer to measure the density or the specific gravity of a liquid food sample, which is related to the concentration of the dissolved substances such as sugars, salts or alcohol. This hydrometry is used in the applications of sugar content measurement, alcohol beverage production, milk and the dairy products. In products like fruit juices, syrups and wines, hydrometer helps to determine the concentration of the sugars, as denser liquids contain more dissolved solids. And in alcohol beverage production, use it to monitor the fermentation by measuring the change in the specific gravity, indicating the conversion of the sugars into alcohol in beer, wine and spirits. In milk and dairy products, it is used to assess the quality and the purity of the milk by measuring its density. It is also used to detect the adulteration in the milk, water dilution, and it is used in measuring the specific gravity or the density, and best applied to the analysis of the solution consists of only one component in a medium of water, that is milk, and commonly used in the beverages, salt, brines and sugar solutions. Examples are pycnometer, hydrometer and Westphal balance. Next is the refractometry. Refractometry in the food analysis uses a refractometer to measure the refractive index of a liquid, which changes based on the concentration of the dissolved substances like sugar, salts and other solutes. It is used to measure the sugar content, that is Brix measurement, commonly used to measure the sugar content in products like fruit juices, honey, syrups. The result is expressed in degrees Brix, indicating the percentage of sugars by weight. And it also helps to ensure consistency and quality in the food products, particularly in beverages, jams and sauces. And it can also detect the adulteration in products like honey, milk by comparing the refractive index to the known standards. It determines the soluble solids in the fruits and the fruit products. When a beam of light that is passed from one medium to another and the density differs, the beam is bent or refracted. Next is the infrared analysis. Principle is it measures absorption of the radiation by molecules in the food. Different functional groups absorb different frequencies of the infrared radiations. For water, near infrared bands, that is from 1400 to 1450, 1920 to 1950 nanometers are the characteristics of the OH stretch of the water molecules. Infrared analysis in food analysis uses infrared light to measure the molecular composition of the food based on how different components absorb IR radiations. And it is quicker to quantify various compounds like moisture, fat, protein and the carbohydrate. It is commonly used to determine the content of these compounds in products like dairy, meats or grains, and detects adulterations or substitutions by comparing the IR spectra of the ingredient. IR analysis provides a rapid and accurate and non-destructive method for assessing the composition and the quality of the food products, making it widely used in routine food testing and the quality assurance. A sample is exposed to the infrared radiations and the decrease in the weight due to the water loss is measured. Choosing the right method depends on the factors like the nature of the sample and required accuracy, speed of analysis and available equipment. Direct methods are generally more accurate but time consuming, while indirect methods offer quicker results but may require calibrations and validation. There are also other methods to determine the water in the different molecular environments, that is vapor pressure method and the thermogravimetric method. This vapor pressure method in moisture analysis measures the vapor pressure of the water in a food sample. to determine its moisture content. This method is based on the principle that the vapor pressure of the water above a food sample correlates with the amount of water present in the food. Vapor pressure methods are used to determine the water activity, which is crucial for understanding the microbial stability, shelf life and the spoilage risk in the foods. And these methods can be indirectly assess the total moisture in the food by relating the vapor pressure to the moisture content. It also helps to optimize drying and storage by elevating how much water remains in the food at the different humidity levels. Determination of the water activity and bound water is less volatile than the free water. The vapor pressure methods are valuable for accurately assess the moisture content and the water activity in the foods, critical for ensuring the product stability and preventing the spoilage. Thermogravimetric analysis. It measures the change in the weight of a sample as it is heated. It provides a moisture profile by identifying the weight loss corresponding to the moisture content. Thermogravimetric analysis in moisture analysis, it's a technique. It is used to measure the change in the food sample's weight as it is heated over time. The weight loss is attributed to evaporation of moisture and other volatile components. It measures the mass of a sample as it is heated at a control rate. Free water normally evaporates at a lower temperature than the bound water. And this thermogravimetric analysis is used to accurately quantify the moisture content by tracking the weight loss due to the water evaporation as the sample is heated. It helps to understand the thermal stability of the food components by identifying the temperatures at which different substances like fats or proteins break down. And it ensures consistent moisture levels in the products like cereals, powders and processed foods. This TGA provides a precise method for measuring the moisture content and analyzing the thermal stability of the food products. It is useful in both quality control and the product formulation. Now coming to the indirect methods for moisture analysis in the food. It utilizes various techniques to estimate the moisture content without direct measurement of the water. These methods are often faster and non-destructive, making them suitable for the real-time monitoring and the quality control in the food processing. Here are some common indirect methods for moisture analysis in the food. Indirect methods estimate the moisture content based on the measurements of other properties that correlates with the moisture content. These methods are generally faster and non-destructive, but they require calibrations. Common indirect methods include the electrical methods. These measures the changes in the electrical properties that occur due to the presence of the moisture in the sample. Next is the near infrared spectroscopy. Near infrared light is absorbed by the water molecules. The resulting spectrum can be used to quantify moisture content after calibration. And microwave moisture measurement measures the interaction of the microwaves with the sample as water content affects the dielectrical properties of the materials. Next is the nuclear magnetic resonance (NMR). It uses magnetic properties of the hydrogen atoms in water to determine the moisture content. With this, we have completed the moisture content analysis in the classical methods of food analysis. If you like the video, please do like, share and subscribe.